In this video, we will discuss about beta-lactam antibiotics, which include penicillins, cephalosporins, cefamycins, and other antibiotics. All these antibiotics contain beta-lactam ring in their structure. Now, let us come to the penicillins. Penicillin was the first antibiotic which is discovered by Alexander Fleming, and that is isolated from the penicillin notatum. And now the commercial production of these penicillins is from the penicillin chrysogenum. And Fleming, Fleury and Chen got Nobel Prize in 1945 because of their work on the penicillin. And the usage and the discovery era of that penicillin is called as golden era in the chemotherapy. And all penicillins have common nucleus, that is 6-amino penicillinic acid, that is 6-APA. Some bacteria have capacity to produce penicillinase enzyme, that is beta-lactamase enzyme, and they have ability to break the penicillin into inactive compound, that is penicillinic acid. They break the beta-lactam ring, and such antibiotics which are sensitive to penicillinase activity, and those are called as beta-lactamase sensitive antibiotics. Now let us come to the mechanism of action of these penicillins. They are the cell wall synthesis inhibitors. They inhibit the synthesis of peptidoglycan in the cell wall, and their binding site was the penicillin binding protein, that is the enzyme that form this cell wall. The penicillin binding protein have transpeptidase and carboxypeptidase activity, and there are several types of penicillin binding proteins, and some of the important ones are listed here. Penicillin binding protein 1A, which helps in the transpeptidation in the peptidoglycan synthesis, and 2 maintains the cell wall shape, and 4 maintains the or carry out the D-alanine carboxypeptidation. The beta-lactam ring of the penicillin inhibit the transpeptidase and carboxypeptidase. Thereby, there is no synthesis of peptidoglycan, and ultimately leading to the death of the bacteria without formation or synthesis of the cell wall. And gram-positive bacteria are more sensitive to these penicillins, and this is because in the cell wall structure of the gram-negative bacteria there is periplasmic space, and in the periplasmic space there is beta-lactamase enzymes are present, and these beta-lactamase enzymes will reduce the penicillin to penicillinic acid, which is inactive. Thereby they are not sensitive to these penicillins. And now let us come to the classification of these penicillins. These are classified as narrow spectrum, broad spectrum, extended spectrum, and last one, that is potentiated penicillins. In narrow spectrum antibiotics, these are again classified as narrow spectrum, beta-lactamase sensitive and resistant. And in beta-lactamase sensitive, these are again classified as acid labile and acid stable. The example for these narrow spectrum beta-lactamase sensitives are penicillin G, which is the natural penicillin obtained from the Penicillium notatum, and which is acid labile, and penicillin V is semisynthetic, which is the acid resistant. Now let us come to the beta lactamase resistant, and these are classified as isoxazolyl penicillins and non-isoxazolyl penicillins. And for example, for the isoxazolyl penicillins, which include cloxacillin and oxacillin, and for non-isoxazolyl penicillins, which include methicillin and neficillin. Now let us come to the broad spectrum antibiotics, which include ampicillin, amoxicillin, and for extended spectrum antibiotics, carbonicillin and ticarcillin. And finally, in potentiated penicillins, as in the case of potentiated sulfonamides, where we combine the sulfonamides with the diaminopyrimidines. Now here we will combine these potentiated penicillins, which mean here we combine penicillins with the beta-lactamase inhibitors, which mean these beta-lactams inhibit the penicillinase or beta-lactamase enzyme, thereby preventing the degradation of penicillin to the penicillinic acid, thereby enhancing the activity of the penicillin. And these include amoxicillins combined with the clavulanic acid, and ampicillin with salbutam, and ticarcillin with clavulanic acid, and piperacillin with tazobactam. And G cannot be given orally because these penicillins, that is naturally occurring penicillin G, is acid labile. Thereby this is destroyed by the gastric acid, which is secreted by the parietal cells of the GIT. And acid-resistant penicillins such as ampicillin, penicillin V, cloxacillin, oxacillin, amoxicillin can be given orally. And the excretion of the penicillins is mainly via kidney, and that is via urine in the unchanged form, and 20% through the glomerular filtration, and remaining 80 from the proximal tubular secretion. The proximal tubular secretion of the penicillins can be inhibited, thereby preventing the excretion of the penicillins via giving the probenecid. This probenecid inhibits the excretion of the penicillins and thereby prolonging the effective blood level of the penicillins. And the dose of these penicillins are usually expressed in the international units.